Good morning, everyone. Welcome back from the coffee break. I hope you've had enough coffee for my talk. So I'm Xiaokang Li. I'm working as a PhD student in BIOS Lab and EPFL here. And today, I'd like to share my history of chatting with the cells by using a label-free nanoplasmonic biosensor, which can enable real-time monitoring of cell secretion. So what is cell secretion? It is a process by which multiple substances are produced and released from a cell. And it's a highly regulated process that incorporates multiple steps from external stimulation to the specific gene expression and the final protein production and exocytosis. And this process concerns a large variety of different kind of molecules, from proteins, hormones, and the neurotransmitters. So such as the antibodies get secreted by B cells, which can fight against the pathogens and protect our human body. And to analyze such a complicated process, typically we can use fluorescence assays such as FACS or colorimetric assays such as ELISA. So here I show a diagram how the sandwich ELISA assay gets done. And it contains multiple steps to put different labels onto the target molecules to amplify the signal for the readout. But although those typical methods are quite sensitive, they can reach like a picogram per milliliter sensitivity, but it has some shortcomings. The main shortcoming is it requires the labels, additional labels onto the target molecules like a fluorophore or enzyme, and it contains multiple steps to wash it, so it's quite time consuming and very tedious. And therefore, if we want to analyze different molecules, you have to put different labels on the molecules. So it greatly limits the scale of the assay. And the most important thing is it only provide us a snapshot readout, which means the result to get is only from a certain time point of the whole process. We lose the information of the dynamics in between. So we need to develop a method that is label-free, which means we don't need to put the additional labels onto the target molecules, so that we can do a high throughput and multiplex biosensing at the same time, and simultaneously. And most importantly, this is real time, which means we can investigate the whole dynamic profiles of the whole process without ending the process at a certain time point. So in our lab, we developed a plasmonic nanohole array biosensor chip for this label-free detection. So this sensor chip only measures like 1 by 1 centimeter. It's quite small. But if you zoom in onto the surface, it is composed of millions of nanoholes that perfectly aligned across a whole surface. And each nanohole is only 200 nanometers and separated by 600 nanometers. So what is the special thing about this chip? Why we can use it for label-free biodetection? If we look at the chip from the side view, so it is a thin layer of gold, metal gold, that deposited on a glass slide. And as we do in the typical method, we can coat a layer of antibodies that can capture the molecules that we are interested. And if we shine a light onto the nanohole sensor, the surface, so what will happen is those nanostructures will greatly confound the light in the volume and enhance the local electromagnetic field and generating optical field for our detection. And on the other side, the transmission light will show a very unique spectrum with a resonance peak here, which is formed by the unique optical phenomenon, the so-called extraordinary optical transmission. And we use this resonance peak, which can be used for the label-free detection. How? So imagine now we have some analyze the molecules that we want to analyze, and they bind on the surface by being captured by the antibodies, and this resonance peak will shift to a longer wavelength. And if we have more analytes coming and bind on the surface, and this peak will shift to even longer wavelengths. So in this way, we're able to see that with the increasing concentration or mass of the molecules binding on the surface, we have a corresponding spectral shift of the resonance peak. And without any additional labels like fluorophores or enzyme, we can do the real-time analysis. So in order to perform such a label-free detection, we implement an optical setup, which is a simple modification of the inverted microscope, where we mount a spectrometer on the side, and we coat the whole microscope with a customized big box for cell incubation. And we call this setup as spectroscopic imaging. So we put our sensor or samples on the microscope stage. We shine the light, and the transmission light get collected by the objective and enters the spectrometer. So what happens in the spectrometer is, first it will form an optical image where we can see our chip or anything on the chip. And in order to precisely extract the spectrum from a certain point, we close the whole image with a slit where you can see is the black part, so that we can zoom in one line on the surface. And from this one line on the surface, we transform this image to a spectral image where we can see all the wavelength information along this line. And then if we select a region of interest on this spectral image, we can extract the spectrum, which we use for the label-free detection. So as a proof of concept study, first I want to demonstrate the feasibility of this chip for VEGF secretion analysis from cancer cells. So VEGF stands for vascular endothelial growth factor. It is a very important factor involved in a process called angiogenesis. And angiogenesis is the process of growth of new blood vessels, and is highly involved in cancer metastasis. How? Imagine we have a tumor tissue or cancer tissue growing in the patient, and if the tumor tissue grows to a certain scale, it needs more nutrient, more oxygen supply. So now, at this moment, the tumor cells will secrete the VEGF, this growth factor, and attract the surrounding blood vessel to grow toward themselves. And once the blood vessels grow into the tumor tissue, the tumor cells can infiltrate into the blood vessels and then travel to the remote places and settle down for the metastasis and grow. So it is a very important factor, and that's why there are some drugs, that anti-cancer drugs, targeting this VEGF growth factor to cure the tumor. So in order to analyze the VEGF secretion, we built here a modular system, which is first the cell module, where we can culture the cancer cells in a winding microchannel design, where you can see here, if we zoom in, the cancer cells, we put them firmly attached on the surface. And then we keep the flow that can transport the secretion onto the next module, which is the detection module, and which is a biosensor chip integrated with another microchannel design of the microfluidics. And here I show that we can pattern 3 sensor arrays in one line, and each sensor array, if we zoom in, is the thousands of nanoholes for the label-free optical detection. And in order to guarantee that our detection is specific to VEGF, not other molecules, so we adopt a well-established surface chemistry strategy on our gold surface. So basically, first we put a layer of thiomolecules which can firmly grasp the gold surface. And on top of it, we have different layers of molecules, protein molecules, which can correctly orient our antibodies for efficient capturing of VEGF molecules. And this surface chemistry can give us quite a good sensitivity for VEGF detection. So here, before the real analysis, we need to calibrate the biosensor by injecting the VEGF of known concentrations onto the sensor surface. As you can see here, with the concentration increasing, we can have an increasing optical signal. And we can detect as low as 200 picogram per milliliter of VEGF. It's quite sensitive. And with this setup, then we put the whole device in our customized microscope design, the spectroscopic imaging setup. And we're able to monitor the whole process for more than 10 hours for long-term analysis under a proper cell culturing conditions with the temperature and with the gas condition supply. And then we can acquire such a data set for the analysis. Like sensor A and B are 2 sensors that coated with the antibodies for the detection. And control sensor is another sensor that we don't have any surface coating as a negative control. So with this data, we can see that control sensor always remain flat, which can show that we have very good selectivity for the detection. And from the other sensor, we can see an onset of the signal around 5.5 hours that shows that the VEGF gets secreted, start to be secreted by the cancer cells. And then after like 1 hour or 30 minutes, we have the final delta lambda, which is the spectral shift, from which we can compute the peak concentration of the VEGF proteins, which is around 240 picograms. And later on, we do an ELISA assay for the same setup, and we get quite equivalent results, which shows that this label-free detection is quite reliable and reproducible. So with this study, we successfully show the feasibility of the plasma nanohole arrays for label-free detection. But then, as you may notice that here, we put like a big group of cancer cells here. A challenge is lots of biological studies have discovered that each cell, even though they share the same genome, the same phenotype, they have their own personality in gene expression, in metabolism, in protein production. And to analyze the personality of each single cell is quite important for our biology understanding, disease diagnosis, and to develop new therapies against cancer and other diseases. So we have shown that our sensor chip can be used for label-free and long-term cell secretion. So is it possible that we can use the same chip for single-cell analysis? We can envision such a setup where we put a single cell directly on our nanohole surface, and surrounding the cells, we can put the antibodies that against the specific molecules we want to detect, and then we shine a light here. And to realize such a setup, we build a device for single-cell isolation and detection. So where we trap one single cell inside a microchamber that's gated by the microfluidic valves, and the volume of this microchamber can measure down to 100 nanoliters. And then we put the antibody around the cell to capture the secretion molecules. But then another challenge comes up that with this such a low volume of liquid under cell culture conditions, which is high temperature, 37 degrees, they can evaporate very fast. So on top of this microchamber, we put another layer of regulation, which is simply a continuous water flow to regulate the humidity and the temperature for reliable analysis. And we adopt a similar strategy for the surface chemistry to guarantee the specificity and selectivity of the bioanalysis. And besides that, we put additional layer of polylysine. It's a polypeptide molecule which can attach the cells on the surface. And this surface chemistry can also give us a quite sensitive detection. And this time, instead of VEGF, we analyze an even smaller molecule, which is called interleukin-2, which is involved in our immune system to regulate the activities of white blood cells. And we can reach also a quite extraordinary sensitivity for IL-2 down to 50 picograms per milliliter. And then we put them in our spectroscopic imaging setup. First, we get an optical image, and where we can zoom into one single cell along this line, and we transform it to the spectral image. And the location where the cell resides, we can see a very specific fingerprint on the spectral image. And then on this image, we can define different region of interest on the cell or some distance away from the cells. From each region of interest, we extract the spectrum for our detection. And I can show here, from one cell, we can get such a signal from different region of interest, from the cell spot, and from the region of interest further away from the cell. And we can see that we get different levels for the signal. Right on the cells, we have the highest signal, and then with the region of interest, they get declining away. And then we can even plot a spatial distribution to better show this diffusion of the molecules along the distance to the cells. And here it's quite interesting that there are some fluctuations on the cell spot, but not on the other region of interest. So in order to show what happens here, we do a real-time live cell imaging, and we observe that the cell will move a little bit within a range of like 5 micrometers, which can induce such fluctuations. But then after that, the cell will stay there and maintain the position. And since this is single-cell analysis, we can do the same analysis to other cells. And here we show another 2-cell analysis with a temporal analysis and with the spatial distribution. And quite interesting here, we see different personality of the cells. So with cell 1 and cell 2, especially cell 2 is quite active, like during the first 2 hours. But cell 3 is very mild, very quiet, it's quite flat. So the distribution is more homogeneous, where cell 2 has a very concentrated distribution and a cell spot. And these 2 studies very perfectly show that the feasibility of our nanoplasma nanohole arrays for label-free detection, even for spatial temporal analysis for single-cell secretion. They get published on lab-on-chip and small paper, and the success shows that our plasmonic nanowhole array biosensor enables long-term and real-time cell analysis and is capable for lab-on-chip integration and also ability for single-cell detection. With these studies, I'd like to first thank all the great support and brainstorming of our BIOS group and my collaborators in Ludwig Cancer Institute and RMIT Australia University for their help with biology and microfluidic fabrication, and in the end, all the funding parties to sponsor my studies. Thank you for your attention.
Thank you, Xiaokang. Questions from the audience?
So I had a question about the spatial distribution that was changing in the beginning for all the cells. Did you try to see if that correlated to any, say, pathway or any response related to the disease of the activity in the initial periods?
To enable the secretion of this molecule, actually we use an artificial chemical stimulus which can activate the cells to secrete. So this stimulus will induce some perturbation inside the cells before the secretion, which could be the movement we see. Yeah, but then different cells, they can respond in different ways. Yeah.
Thanks.
Thank you for your presentation. I'm wondering about the fabrication of the chip.
Yeah.
With the beautiful nanopore.
Nanopore, yeah. The fabrication is, we can use E-beam or deep UV lithography. So the advantage of deep UV lithography is we can pattern the nanoholes over the whole wafer scale, yeah, for a large scale production. With E-beam, it's time consuming and you need to do point by point. Yeah.
Nice presentation.
Thank you.
So compared to the in vivo microenvironment and the secretion profile of cancer cells over there, do you expect a deviation on your chip because of the mechanics and also because of optics? Like the fact that you have laser beams and energy provided into the system may change the temperature, maybe. So can you comment on the environment that you create and the implications for secretion?
So first answer the question about the light source. We didn't use laser. We used the halogen lab to provide a broadband illumination. And later on we used the LED source, but we are afraid that laser may be too powerful to induce any temperature change or the harm to the cells. And in terms of microenvironment, yes, actually we are studying on that. So here we only put the cells there without any support of the stroma of the microenvironment. But we had a recent study that we create a biomimicking surface on the gold plasmonic arrays by putting a lipid bilayer to mimic a cell membrane. And then the cells, they can more behave like in vivo microenvironment. Yeah, that is the ongoing research we are doing. Thank you.
We have time for more questions. If not, I may introduce a little remark on the microengineering part, since there was a question about this. We are very specialized in this at EPFL. We have a center for micro and nano fabrication, which provides a lot of facilities to produce these type of elements and cross-disciplinarity between different fields of research. Okay, let's thank Xiaokang Li once more.